Good afternoon. My name is Fiorella Gurrieri. I am a medical geneticist from Rome, Italy. I work with the research group since 2010 on AHC. We work at the Catholic University of Rome at the Institute of Genomic Medicine. We have been involved both in the clinical research, meaning patient description and identification of specific phenotypic features, and also on basic science. We have been involved with the US group on the gene identification with Dr. Einstein and Goldstein and all the group. And then we moved on to the identification of disease mechanism at the cellular level. Now we have some cell lines from neuroblastoma human cells in which we have introduced the constructs, the abnormal genes, with the 5 most frequent mutations in ATP1A3, and we are studying what happens to these cells. And what happens is that the cells show a number of alterations, such as accumulating sodium, for instance, and some others, morphological alterations. And we use these as markers to understand when any drug can rescue the phenotype. Let's say can clean out the sodium from the cells. So we have an agreement with a private company, pharmaceutical company, to screen about 2,000 compounds. Most of them are safe in men, which means that are compounds that can be redirected easily. And so we want to see whether we can find any of this drug that can rescue the phenotype on our cellular model. And so this means that we need a lot of people involved. And in fact, we have a group, a small group actually. Danilo Tiziano and Agnese Novelli, Emanuela Biusi, Serena Spartano are the people who are working on this, and we have a collaboration with another Italian group of chemical pharmaceuticals at the University La Sapienza, Professor Ragno, and they are doing crystallographic modeling of ATP1A3 in order to screen compounds that can interact with ATP1A3, wild type and mutated. Also, we could not do this without the very good support of the Italian Association, both in terms of funding and also in terms of organization, and even we owe them our presence here at this meeting. We are currently doing this research. We are at the very point where we are starting the screening, the drug screening. We have set up a number of cellular characteristics, and now we are going to screen the compounds. We already have the compounds from the company, and we hope we can have results maybe by the next symposium, hopefully. We think we need a lot of collaboration. For instance, we have already found a collaboration with Professor Mikati, because as soon as we have compounds that are helpful in cells, we can try them on the mouse model. And this is an important step, of course, because he has the most severe mutation in mice, and so we can try to screen that. Professor Mikati has had a number of suggestions on what to do in addition to better characterize our disease model in the cells, which is to study a bit deeper the ATPase function. And we are trying to do this. We really have a good support from the Italian Association of Families. But of course, this is not enough, and we may need more. But we really would like to be part of a network with the same goal of the treatment, and in which everybody can do something. And so we really think that collaboration is crucial in this specific condition, because it puts together different competences of people, different knowledges, backgrounds. Everybody's important in this research, we think. What we may need in case some of this works, and we hope so, then we need funding to go on, because to go from the simple screening to additional screening and then to go to patients, we need a bit more commitment and funding. We do like this symposia, the ATP1A3 symposia, because they bring people and families together. Maybe I would like to suggest to have even more involvement of everybody, which means that maybe sessions can be organized as round tables, where let's say 10 people working on the same little field within ATP1A3 can talk to each other and exchange experiments, experience, and ideas, because I think we really help growing.